Welcome, everybody, to the Fibrolamellar Cancer Foundation's Patient and Caregiver Education Series. I'm John Hopper, president of the foundation, and I'm pleased tonight to introduce a topic focused on clinical trials and future treatments, featuring a very distinguished panel of oncologists. For those of you who are new to the series, this is just one part of the foundation's three-pronged mission: to find a cure and treatment options, to raise awareness of this disease, and to connect and support our community of patients and their families. The series addresses key informational needs of the community through a variety of topics and formats. For a listing of past and upcoming programs, please go to our website at fibrofoundation.org and click on the Resources and Events menu tab. There are programs scheduled throughout the year, including topics such as immunotherapy, patient care resources, caregiver to caregiver fireside chats, and much more. If you have any questions tonight, please put them in the Q&A located at the bottom of your screen, and our scientific director, Dr. Mark Firth, will raise as many of these to our panelists as time permits. I always have to make a notation when we do this that the foundation does not give medical advice. So now I want to introduce our panel host, the distinguished Professor Ghassan Abou-Alfa, MD, from Memorial Sloan Kettering Cancer Center in New York. A world-known expert in liver and bile duct cancers with significant interest and focus on fibrolamellar carcinoma. In fact, Professor Abou-Alfa has been hand in hand with the foundation, I think, Ghassan, since day zero almost, since I believe you helped to treat our founder, Tucker Davis. Okay, with that introduction, Ghassan, I'm going to pass the baton to you.
Thanks so much, John, and hello, everybody. Good evening and good morning, wherever you are, and good night, wherever also you are, because we hear that and we see that you are from everywhere, and we're happy to welcome you all. It's my great honor to help lead on the discussion today, and if anything, I'm very pleased and super excited to be joined by my very dear colleagues, and I'll start by introducing them. First, we have Professor Allison O'Neill. Dr. O'Neill is the clinical director of the Dana-Farber Cancer Institute for the Solid Tumor Program, medical director of the Dana-Farber Cancer Institute Boston Children's Hospital Tumor Center of Excellence, and the chair of the COG Liver Tumor Committee. Ali has been heavily involved in regard to the care for patients with fibrolamellar carcinoma, and I'm sure we're going to hear quite a bit from her. I also would like to welcome our very dear friend, and I have to admit that he became a dear friend because of fibrolamellar, Dr. Paul Kent. Dr. Kent is from the Rush University Medical Group. Super exciting to always hear him, and I really would always admire his vigorousness in regard to the disease as a pediatric hematologist oncologist. I'm also joined by Professor Ahmed Kaseb. Dr. Kaseb is a professor in the Department of GI and Medical Oncology at the Division of Cancer Medicine at University of Texas, MD Anderson. Dr. Kaseb has been with us in the fibrolamellar war from really the start. And lastly, we have our very dear friend and a great colleague, Assistant Professor Dr. Mark Yarchoan, who is, again, led a quite innovative perspective in regard to fibrolamellar carcinoma, and Dr. Yarchoan joins us from Hopkins in Baltimore. With this said, I'm gonna trigger some of the discussion. As you noticed, we are not here to lecture, we're not here to kind of, give any specifics, but we're here to really learn from each other. So please just imagine we are in the virtual world nowadays, but think about all of us who are sitting around like a fireplace and we're just chatting about what we think is best for our patients and how we can help best in regard to the service that we can provide to all of the patients with fibrolamellar carcinoma. And with this said, I just kind of would like to pick up the discussion first. And if anything, I'll ask Dr. Kent. So Paul, for all of us, teach us, like fibrolamellar carcinoma, we understand does not have a standard of care. So we kind of like depend on something called clinical trials. What's that?
Yeah, thank you. So a clinical trial is a way to establish what is the best care, what is the most successful or the most safe and efficacious treatment in a very disciplined scientific manner. We start with what's called a phase 1 trial. It might be very small, 10 or 20 patients. It's something that seems promising in the lab, maybe promising in animals, and we're really just looking at whether it's safe. Then the next step comes phase 2, where it's a slightly bigger. We're trying to say, well, which diseases might it most be helpful in? And can we try to, now we've established a dose that seems to be safe from phase 1. Now we're in phase 2. Let's see if we can figure out what diseases it might be helpful with. But then the golden, the dream is to get to the phase 3. A phase 3 trial is where you're trying to compare your new idea, your new experimental treatment with what's considered the standard of care or the control group. And that's really what is considered the ultimate evidence when you're speaking to doctors about what is evidence for medicine. Do we have a phase 3 trial that says the new idea, the experimental idea is better than what's considered the standard of care? Now, as you can imagine, this is quite challenging when you have a very, very rare disease. The only way to do this is lots of cooperation. Everyone's only has 1 or 2 patients. How do we get all that data together? How do we collect all that together and really learn something about what is the best treatment for somebody? You're going to have to collaborate. You're going to have to work together.
That's very nice. Thank you so much, Paul, for that. And along that line, I'll pick up on a few of the points you brought up. And of course, we'll engage other of our colleagues. So Ahmed, Paul just mentioned about clinical trials in different phases. And he also mentioned, which we all agree, that thankfully, the cancer, fibrolamellar carcinoma, is relatively rare, thank God. But at the same time, we do understand that we need to practically get to certain answers. So the phase 3 trial that Paul just brought up, which is technically is randomizing against a placebo or a certain standard of care, which we don't have anyway. Do you think it's going to be feasible? Or if not, how can we get to a certain answer based on those clinical trials?
So this is a very critical question, and it's peculiar not only to fibrolamellar hepatocellular carcinoma, but to all rare tumors. And as you know, Ghassan and Mark and Ali and everybody, that nowadays even common tumors have rare entities among them. So this is something that we're going to have to know how to deal with going forward. So for example, in rare tumors, we could, put our efforts together into smaller studies, even if it is randomized, could be randomized phase 2 rather than phase 3. However, the main challenge we have with fibrolamellar now is that we don't even have a standard of care regimen yet. So I think we're all trying from, each one from, his own angle and our own team efforts to come up with something in a pilot fashion. But eventually, we really have to put our efforts together to conduct larger studies, either to establish the first standard of care or to compare two regimens. So it is, a race to the benefit of our patients.
Thanks so much, Ahmed. I'm going to hold on to another important point that Dr. Kent brought up, and I'll come back to it, which is about how can we work all together because of the rarity of disease and how to collect to those studies, which already you referred to as well. But before we go there, I would like to actually ask Dr. O'Neill, as a pediatric intelligence oncologist, same like Dr. Kent, but in general, the perception that we have is that the pediatric colleagues do way better in regard to accrual to clinical trials compared to the adult colleagues. Tell us a little bit about your secret.
Our secret, it's a good question. I think it's because everything we treat is rare. I mean, the reality is, and this is a thankful reality in the pediatric space, is that cancer in children is rare. And so everything we study, we have to study not only with rigor so that we can arrive at the right conclusions, understand in a uniform way what we're studying, and make adequate conclusions regarding whether the drugs we're studying work. But we really have to collaborate because the numbers are so small that we really can't learn from individual diseases unless we study them in a consortium-based way or we align institutions towards a unified goal. So I think when we think about a disease like fibrolamellar, HCC, we are so well positioned as pediatricians and pediatric oncologists to think in that manner because we're so used to considering the best and most rigorous and effective way to study rare indications that that approach lends itself nicely to the study of fibrolamellar carcinoma for that very reason.
Fair enough. All what I can tell you, I can't wait until I see a big study, and we're gonna call you to name it something, because after my favorite study, I know they're not favorite drugs, but the Play-Doh that you guys have in pediatrics really excite me quite a bit in regard to the name. So good for you. So Mark, to be fair to our patients, can you please help us walk any of our patients through the clinical trial? In other words, imagine that a patient comes to you to the clinic at Hopkins, and you're gonna try to set them up for a clinical trial. Generically, in other words, what's the process? Like, what are the things that you go through? What are the processes, and what are the challenges and the fears from the patient perspective, if you can?
Yeah, there's a lot to untangle there. I'll try not to take too much time here, but
take as much as time as you want. We have plenty. Yeah, don't worry. I'll blame John if we're behind.
Maybe we could just start by, really defining exactly what it is to be on a clinical trial. There are different kinds of clinical trials, but normally, at least in our world, a clinical trial involves some kind of treatment or intervention, like a new medicine or chemotherapy for fibrolamellar cancer. And so, before a trial is put together, you need to have some sort of roadmap for how the trial is going to take place. And this is called the protocol. And so those of us who run clinical trials, we spend a lot of time writing exactly what the treatment is going to be, how it's going to be given, how it's going to be monitored, what labs we need to collect, and these kinds of things, and exactly what it is that we're going to measure. If it's with CT scans, what time are we going to measure these CT scans? So when a patient comes in for a clinical trial, and they're interested in that trial, the roadmap for how they're going to be treated is already made up. It's not something that we make up on the fly. It's already set up and written down on paper. And that roadmap is actually approved by sometimes an IRB, which oversees the trial. And sometimes
and
even
IRB means Institutional Research Board. It's like a bunch of doctors, and sometimes there's community people that will speak on or advocate for the patients that sit together to decide that this is okay, it's not hurting, it's helping, correct?
Exactly. So the patients may not realize this, but when they come in to participate in a clinical trial, there's often a year or years of work just to set up the trial with the exact procedure of what's going to be done, with a whole team of doctors, not just the doctor they're seeing, who oversee the trial, make sure that the trial is safe and reasonable to conduct, and make sure that the trial is being conducted correctly. So I think that's the first thing I would just say, is that this is a major operation, and that patients often only see the tip of the iceberg. There's a data manager, there's a nurse, there's a team of doctors who are overseeing the trial. There's laboratory people sometimes who are involved in studying the trial. But patients don't see all that. So from the patient's perspective, they come in, and normally the first visit, at least here, the first visit is normally to talk about what the trial is. That trial, the information visit, we sort of discuss the pros and cons of participating in the trial. And there's something called a consent form, which really lists exactly what's going to be done, what the risks of the trial are, what the potential benefits of the trial are. And that's an opportunity for patients to really make sure that they understand what it is that they're signing up for, and what their alternatives are. What else could they do instead of the trial?
Let's hold off here. You bring a very important subject, which is the next anyway, so perfect timing for that.
Great.
So again, I would like to, however, reiterate one more time for all our listeners and people who are watching this, that as you said, the trial has a lot of complexity behind it, but we need to assure everybody they don't need to worry about it. The way I jokingly say to the patient, just follow the instructions. We'll tell you exactly what to do, where to do, which floor, which door, just do what we kind of tell you to do, and don't worry about the details behind that. So this, I totally agree with you. This is very important for the patients to know about the simplicity of doing things from the operation of the patients. And as I said, and you already referred to on the IRB, there is always members of the community that represent patients in a way to see things from the eyes of the patients and the loved ones, to make sure that this is reasonable, appropriate, applicable, doable, etc. Then you bring in an important component about the consent. And I would like to go back to Dr. Kent. I would like him to comment on what is a consent is. And then because also, Paul, you are in the pediatrics, teach us about what's in assent.
Consent is really sort of documentation that you've had an honest conversation about the risks and benefits. It's a common misconception that a consent is a legal form. It has no legal bearing, but that's a common, but it's really meant to be a way to show both to you and to the patient, both of you, that you've really discussed everything, that you've discussed all the risks, all the benefits, what's known, what's unknown, what is expected, not expected, what are the expectations of you, the person running the clinical trial, what are the expectations of the patient. So that's the first step, to make sure if someone has had, and the 3 golden rules is that you have enough information, that you're free of any coercion, and that there's no pressure that being supplied in any way, so that you have a completely full ability to say yes or no, and it's clear to you that you'll get the same level of care whether you agree to the clinical trial or not. And assent is complex. It has to do with acknowledging that a child should have an opinion in the process. Now, of course, that depends on the opinion, on the age of the child and the maturity of the child. But the general rule is the so-called rule of sevens. Age 0 to 7, for the most part, the child has no participation in the assent or consent process. From 8 to 13, they do have a very simple process, simply to say, Dr. Abou-Alfa or Dr. Kent or Dr. O'Neill has explained to me and answered my questions and have talked to me directly. And then from age 14 to 17, it's almost not quite, but almost like a real consent, that they've had all their questions answered and they have a chance to sign and they have a chance to say yes or no. And it's acknowledging that patients in that age group have some degree of autonomy and can't completely be the decisions made by their parents. It's another way to show respect for the person. That's really what it's about, to show that respect, that you have spoken directly to the child and shown them a level of respect, that it's their body and you're the one that is, you're not just ignoring them and speaking to the parents. But it's an essential piece, and this, it can be a long process. It can be a difficult process. But when we remember that this all came down from the Nuremberg trial, we understand why this is important, that there's absolute transparency, that this is fair, that you have a right to choose or not choose, that there's no coercion, and there's what we call equipoise. Equipoise is a, pretentious word that simply means, as far as we can tell, the choices are equal. We're not giving you one choice that's bad and one choice that is better.
Well, I very much appreciate this beautiful description of all of that. And it's very important, especially, I would like to again reiterate what you said perfectly well, Paul, is that this is not a legal document. In other words, we are not contracting between patient and physicians in regard to any clinical trial. The consent, exactly as Dr. Kent said, is simply a iteration of a discussion that has happened. I informed you about what the trial is. You heard about the trial is. I answered the questions. You are really well aware of the discussion. Interestingly, the verbal consent is way more important even than the signature. Signature is only for memory. It's not really for any other reason. And as we already said, you can sign a consent. And let's say you drive off the street and say, you know what, I don't like to be here. You can simply just call and say, you know what, forget it all about, and you can have the full right to do so. So that's very, very important.
With no harm, no consequences.
Exactly.
This is not for me. Yeah.
Exactly. And the other thing you bring in is the issue of the assent. And this is really where it's important for the families, especially for the young patients, is that to be aware that, yes, we engage with every patient with an understanding that they have regard to disease, understandably with the respect to where they are in regard to perception, but definitely the family will be involved as well. Nothing will be involved except with the agreement with, you know, the caring family member or adult for patients who are minor. With this said, I would like to touch on a very important component of the trial, and I'm going to go to Dr. Kaseb for that. So Ahmed, I'm really scared. I read this consent form and oh my God, like the side effects are like scaring the hell out of me. And there's no way I'll freak out even to consider this. Tell me a little bit more. Why are you telling me that I should go on that therapy while the side effects are like super scary and you're making me read all those pages of side effect after side effects? Tell me more.
Yeah, of course, and that's very, very valid concern here. And we take this very seriously because most of the time our patients are young and haven't been through any of these, discussions before. So in general, we go through the consent and through the drug information almost line by line. So we also include our multidisciplinary team, like any other institution where you can have the pharmacists also give their perspective. So in general, what it is, we go through the available evidence of each drug, if there is any available evidence, even from retrospective data, for example. We also rely on data from the larger studies from the parent disease, hepatocellular carcinoma, to give them, to give the patients more input if these drugs were tested in hepatocellular carcinoma. And then we go down to the granular details about the drugs. What are those side effects? What is the percentage? And a lot of times you find out in the those handouts, the percentage, which is really important. So if it is less than 1% or less than 5%, and those are the scary ones, then we have to really talk to our patients about, the chances of this happening in their age, and also given any comorbidities. So it is a discussion that really is critical to have with the oncologist to talk about my specific case, my specific comorbidities and age and everything else. What are my chances of suffering those side effects? And the next important question is, what if one of these side effects happen? So we walk, our patients again, like all of us were trained to do this, regarding how to manage those side effects. How bad are they gonna be in terms of affecting their quality of life? What is most critical to you as a person in terms of those side effects? I've had some patients who tell me, I don't want anything oral. I'd rather have some IV drugs. And I've had patients who say the opposite. So all of these things weigh in our decision because it's their decision in the end. Our job as physicians is to talk to them about the pros and cons of each approach, what we think would be best for them. However, we always have to take this mutual decision with our patients based on their specific case, quality of life, and based on their own priorities as well.
Thank you so much, Ahmed. This is very, very helpful. And if anything, can I clarify one point that you brought up, that the adverse events that are spelled out are not necessarily that they will happen, but we're obligated to spell out everything in case it happens. And if anything, always good idea to check with your doctor, check with your medical team, because they can guide you accordingly. Make sense?
Right. Makes perfect sense.
Perfect. Great. So I'm going to re-summarize all what we heard so far, because we're going to go to another very critical subject. But again, to reiterate, we spoke about the different phases of clinical trials. We said that within the context of the fibrolamellar carcinoma, clinical trials might be needed for only a specific purposes of understanding if there's disease benefit. There's also a concern for the need for the benefit from understanding if it's going to be safe. But we know very well that comparison might be a little bit difficult because of the limited number of patients. And as such, we might be limited to a phase 2 or phase 1 clinical trial, as Dr. Kent brought up. Number two, we spoke quite a bit about the importance of the process of the clinical trial and what will be important to understand that, yes, there is behind the scene a lot of complexities that occur, as Dr. Yarchoan brought up, but at the same time, we do acknowledge that the functionality of the trial is rather very practical, and from the patient perspective, it's really just follow the instructions and you'll be all set and we'll always take care of you very well. Then I kind of bring up an important component in regard to the consenting and assenting. And we agreed that it's not a contractual thing, but it's rather only a opportunity to ensure that we explain ourselves, that patients' questions were answered, and accordingly, this is more of a commemoration of the discussion, but it's not a legal obligation in any way, shape, or form. And as Dr. Kent said, you can just walk from it anytime you want. The other thing we just spoke about is the adverse events that can occur with the clinical trials, what we call in other words side effects. The list usually are long. Please, we ask you all not to be scared of those. If anything, they really can be implemented in the trial because we're obligated to mention all. But at the same time, of course, always patients and loved ones are encouraged at all times to call. And actually, we're all very well trained to make sure that in the start of a certain trial, always we tell patients, please call us anytime. We understand this is new to you. We understand things might evolve, and we're definitely always there. At worst, we're going to tell you, don't worry, it's okay. And at best, we'll at least try to help you to alleviate any symptoms. And lastly, I go back to a point that was brought up in regard to the importance of the collaboration. And I like to focus quite a bit on what Dr. O'Neill mentioned in regard to the pediatric population and the rarity of the diseases. And that's why the collaboration is very key. And to be fair, I also will second what both Dr. Kaseb and Dr. Kent brought up in that regard, which is collaboration among us is definitely very critical. Please be assured, we're all sitting here because we're really very dear colleagues to each other. And be assured, you might travel from one to the other or see different opinions, what have we, but you'll be amazed how much we talk to each other. We are really readily available to each other at all times. And at the end of the day, we all see it. No matter whose patient it is patient as if it's any of us, and we'll help each other at all times to make sure that we do the best. We do this out of passion and out of compassion and out of care. So with this said, and I'm sure we're going to get a lot of questions on that part, but I would like to go back to fibrolamellar, which is the key element that we're discussing and the focus of that disease. And my question is there any possibility that the trials that we do are going to be based on science? In other words, something that we think about or we know about, or how do you come up with the trial? And Dr. Kaseb, we'll start with you because you've been involved from day zero.
Yeah, so typically this is exactly how we would like things to go to, and we call it from lab to clinic and back from clinic to lab. So in general, how these drugs come about for cancer patients, they start from the lab in a cancer cell culture plate and then animal model. And then eventually we take this to phase 1, which means a very initial first in human study to make sure it is safe. And then we take it to the next level, which is phase 2, to make sure that there is a signal efficacy activity. And then phase 3 would launch the drug as the standard of care. So for rare tumors, sometimes it is hard to even find the right research model, in the lab, either in cell lines or animal model that could really be a good fit for patients to test those drugs. And so we have both models going on. We have a lot of clinical trials in the US and abroad, which are based on both of these models, but most of them are emerging from established drugs that have been tested in other cancers, also specifically hepatocellular carcinoma. So that's, what we usually rely on, either preclinical evidence of the activity in the specific disease, or in rare tumors, if it is hard to go through that route, is to start with those drugs that are available for other tumors with a signal and a rationale to use in this specific rare entity. For example, testing the tissues for a specific marker, and then we have start a trial using a drug against this protein or this receptor, for example. So this is what we have currently in the field. Also, a similar concept is to try a regimen and learn from it, and then based on the experience with this regimen, you try to modify it to take it to a clinical trial. So all of these concepts have been tested and have been around, and most of the clinical trials that are currently ongoing are based on this later model.
Okay, within that context, though, I would like to go one step even earlier than that. And Mark, again, you've been involved in quite a bit of work. How do you talk to the guys in the lab? Like, in other words, do they just pick up the phone and say, I have a great drug, let's do a clinical trial? How does this interaction happen?
Well, in my case, I still have a toe in the lab, and then I run a lab. And so for me, right now, my office here is right next to my lab across the hallway. But, you know, I think there's this constant exchange of ideas between clinicians and laboratory folks. And I think that's really important, especially for a rare cancer like this.
And do they, in other words, and yourself in the lab, how is the process? Like, how does something, is an experiment become a drug? Like, what happens there?
Yeah, I mean, you know, for a cancer like fibrolamellar, a lot of the drugs that we study were drugs really developed for other cancers. And then we move them to fibrolamellar cancer. And so many times there's this back and forth between the lab and the clinic many times before it gets into fibrolamellar cancer, as I think Ahmed said. You know, the average time between when something is in a petri dish and when it goes into people is about 10 years. So this is a major long process. It goes into petri dishes, then it goes into mice, then it eventually goes into people. So this is a very long process. You know, for me, the most exciting thing is taking a drug that has shown process and success in other cancers and bringing it to patients with fibrolamellar cancer. I think you're muted, Ghassan.
Sorry, and can you tell us, please, a little bit, Mark, about like how many drugs get to be tested in the lab for one of them to get successful in the, in other words, how much of an effort is this? Like 1 out of 10, 1 out of 100, 1 of 1,000, 1 of 10,000, your number?
Yeah, I mean, you know, the latest numbers are that it takes about 10 years and it takes about $1 billion to develop a drug for cancer. So that's the latest. And but many of the drugs that are developed now, or the approaches that are developed now, if they work in one cancer, they end up working in many cancers. And I'll just give you an example, which is these drugs, these immune checkpoint inhibitor drugs. You know, they were studied in mice for many years, and then they were studied in melanoma, where they showed some success. And then once there was a success in one cancer, these drugs got tested in many, many different cancers. And so, you know, before people say, oh my gosh, we're never going to get a drug because 10 years, $1 billion, that seems like very long odds for fibrolamellar. I think we're going to be the beneficiaries of drugs that are developed for other cancers. And so a major effort is to figure out, with all this work going on for so many cancers, which drugs should we bring to fibrolamellar cancer and to offer our patients, or which approaches should we bring to our patients?
Fair enough. So Dr. Kaseb, so with this kind of, you know, figure that Dr. Yarchoan is bringing up, which means it's really a lot of work that take forever, how can we help speed this up? I presume, and I'm not trying to put any answer here, but I presume it's about like the more we do and the more we get patients involved, the better, correct?
Yes, that's very, very true. So getting our patients involved is critical. not only, to run them through our clinical trial portfolios, at different institutions, and try to also help push those trials to move faster, but also involving our patients with the processes is very critical for us to understand what are their main concerns in terms of quality of life. And in designing those clinical trials, we really have to put this into consideration. What are our patients' population comorbidities look like? What are these patients worried about the most? These agents some of them are more toxic than others. In the meantime, our patients' openness, for example, to participate in research and subject their tumors to biopsies could help the field. And understanding the critical importance of clinical trials reliability, we rely on those sampling from tissues and from blood, for example, to understand the tumors better and design the next best clinical trial. So our patients' involvement is critical to our understanding and their understanding of how they could help move the field faster through all of these maneuvers.
I like that. So now I'm gonna spice it up a little bit. And Paul, I'll have to ask you this because I know it's a dear subject to you. I have to give you credit. You are very good at this, and you really kind of, led quite a bit of effort with that. But why should we care about clinical trials? Why don't we do things retrospectively? Your thoughts?
Yeah, no, that's also a very fair question.
I'm talking to Paul, not you. Except you want to change your name to Dr. Paul Kemp.
Well, clinical trials are the ultimate evidence when we talk about evidence-based medicine. And of course, evidence-based medicine is how we all want to practice. And it goes to the heart of the do no harm concept of medicine. And is it safe? Is it efficacious? The problem, as you pointed out, is twofold. The delay, people waiting and waiting until you have something that's proven. And the other problem is accessibility. How easy is it for you to get this new promising clinical trial availability? And I guess the third would be applicability. Is it a single drug? Is it something that will give important scientific information but doesn't seem likely to help? So then you're stuck in this space where you don't have the rigid science that we all want, the rigorous science what we want, but we also have patients who are going to die and are sick and need help right now. So then you're in a difficult position, because is it more harmful to do nothing because we really don't know what the right treatment is, or is it more harmful to do something that seems reasonable, that seems to have some scientific validity? And that's a tough call, no question about it. So I think that's a position that happens frequently, actually. What do we do if this patient is not eligible for this trial or that trial, or they're not a good choice, or it's not convenient or even plausible for them, never mind the insurance restrictions and transportation and everything else? What do you do then? And your choices are to say, well, I guess I'll do nothing because there's nothing really proven, and I don't want to take a risk, or say, well, doing nothing is also a risk. Maybe we'll try something that seems reasonable, that has some scientific validity, but we'll have to watch you very closely. And that data, called retrospective data, is not as convincing as prospective data, no question, and certainly not as well controlled as a clinical trial. But there has to be something in that gap between the here and now patients who are sick today and where we want to get to, which is this rigid scientific proof that we consider scientific proof, phase 3 clinical trial with a randomized control group.
Okay, I like that. This is very, very good assessment of, understandably, we can have limitation. We have actually an audience with international patients. Not everybody's have a access trial to them. And there could be other limitations here or there. And as such, yes, you might come with that. But you bring a very important other point, and I would like to touch on with Dr. O'Neill on that. So, Ali, retrospective data, what does it mean to you scientifically, and how can we benefit from it, if at all, for ultimately a standard of care, if we can?
Almost like you read my mind, because I think that's a really important aspect to discuss for patients and families when they're thinking about how to understand the historical context of things that have been used in the past, and how to understand the way we think about clinical trials and in following patients prospectively or in real time as they're getting treated and going forth. I think that there is something to be said for looking historically. So for instance, and this harkens again to the importance of collaboration. You know, I think we as individual centers tend to rely on very different go-to therapies because there isn't a uniform standard. And I think what we've learned, particularly many of us engaged in the Fibrolamellar Foundation, is that as we speak to more of our colleagues, as we engage with each other on a more regular basis, I think we find in the pediatric world, we tend to use different agents perhaps than our adult colleagues. And I think that eventually we wind up in the same place, but that's only after multiple regimens or going through multiple regimens with our patients. So I think from a retrospective perspective, and just for the folks on the call, that means looking back in time, looking at what we did over the last 10 years. It's very helpful for us to all align, to collectively look at our data and to say, hey, I treated X number of patients with this regimen. What did you do? You've treated X number of patients with this regimen. And if we have enough overlapping evidence or overlapping data, we can start to draw some conclusions and some patterns and better understand which perhaps treatment backbone makes the most sense upon which to base study going forward. But I think, you know, going back to my point about retrospective versus prospective, I think as we treat patients with new regimens or we take drugs that would have been used for other indications and try them in fibrolamellar patients, it's so very important for us to collect that data prospectively and in an organized fashion, such that we're not left with going back in time and looking to see how those patients did in a less organized fashion. You're always at the risk of error or bias when you look back on data and you look back on patient course. And it's really only in the context of rigorous prospective study, enrolling patients on a trial so that you know every single detail of what happened in real time, that you can reach the appropriate conclusion. Because even the smartest physicians, even the smartest consortia or group of physicians, we are certainly subject to bias when we look back in time at data. So prospectively following our patients is really the gold standard, if at all possible.
Great, thank you so much, Ali. This is a very, very important. And again, it really put things in perspectives as kindly Paul brought it up and you dissected further. So this brings us to the last point that I would like to bring in with Dr. Yarchoan. I'm aspiring for having a collaborative clinical trial and you're going to be leading it. How do you do that?
Well, you know, we have to learn from every patient with this rare cancer because it's the only way that we're going to make progress. And I think, you know, the dream, looking at this panel of esteemed experts here, is that we're going to tackle this together as a group and learn from every single patient that we collectively treat. And, you know, my dream is that we would have studies open at all of our centers so that we can evaluate different treatments together. And I think, you know, forums like this are great ways that we can start to work together and come up with ideas. It's going to take us a while to get there, but already, you know, you've been involved, Ghassan, in some multi-center evaluations of different therapies, and I think that's something we need to build on.
Fair. And we have to really at least give a little bit of taste to our patients about that. It's not like we don't like to work together. We are very good about working together. And really, we kind of like, because of the enormous and importance of fibrolamellar carcinoma, and because it touches many of our dear patients at young age, it really appeals a lot to us, and we see patients before we see anything else. However, there's a lot of complexity in regard to how to do the process that we have to be well aware of. We have, I know, some audience from Europe, and to give an example, for example, just the legality of how you do clinical trial, if it is in Europe versus in the US, it has another dimension. Because, for example, in Europe, there is a right for the patient in regard to right to control the data they have, even the right to forget. In other words, you really want to get rid of the trial, you are obligated to take all of your data out. In the US, it doesn't exist. And we have to really be very aware of all those dimensions, be able to really build a clinical trial. So it's not as simple as it might be. So please, if you don't see us yet working together, it's not because we don't like to, we love to, but because we have to really have lawyers involved. And you know, the minute you have lawyers involved, it takes a little bit longer time. They do very great work, but unfortunately, they have to really kind of, tighten every kind of, screw correctly to make sure that we get where we are. So with this said, I'm going to re-summarize what we just heard about the process of the trial per se. And we heard very clearly that the process starts with the patient coming on, having certain requirements needed. We heard also about the way the process of the trial evolved, as Dr. Yarchoan brought in that it might start with an idea. It makes longer than needed sometimes. They just evolve all the time. The good news is, and I would like to read a second that of Dr. Yarchoan, exactly, he exemplified it with himself, but we all actually have good connection with all our scientist colleagues, or even some of us have labs that really we just build or experiment things ourselves to hopefully be able to bring it to the patients. We spoke about a very important component about the retrospective analysis, because not always trials are available. Yes, we have to be flexible. Yes, we have to be practical. We have to be very flexible, and because we have to respect that not everything is accessible. But if anything, with this very important perspective that Dr. Kent brought in, Dr. Neil warned us again, and Dr. Kent, to be fair, he brought us as well this: we cannot claim anything. I cannot just go to the FDA and tell them, hey, by the way, I have this analysis that I did retrospectively. We need to control a certain clinical trial to be able to claim something is valuable. Because remember, the patient on the clinical trial are doing this on behalf of all other patients. And when we experiment with a compilation of patients, it means that we're going to be able to claim that this is accessible and doable for patients everywhere out of the clinical trial. Same like it's happening nowadays with many things. I think we all are hearing about clinical trials with the vaccine and what have we. So it's kind of like something along that line. And if anything, we heard a lot from Dr. Kaseb, how it's very important to evolve the idea and to kind of like see where it's going to fit in regard to what stage it is, in regard to which phase of the study it is. And lastly, Dr. Yarchoan brought in a very important component, which we all spoke about one way or the other, about the collaborative effort that we need to really be able to bring a clinical trial to fruition and be able to benefit our patient with it. Even though we did acknowledge the certain challenges that we might face, and we just have to continue to work on those and try to circumvent them everywhere we can. With this said, I know that we started 10 minutes early, if I'm not mistaken. We are 8 minutes from the scheduled time, but I'll pass it on to Dr. Mark Firth, dear colleague. We have great admiration, and Mark has been an instrumental scientist in the foundation, and Mark is going to help us lead the panel for the questions. So Mark, back to you.
Thanks, Ghassan, and thank you, Ghassan, and thanks to all the panelists and very much to everybody who's in attendance. I'm going to start with the first question that really I think is germane to a lot of the building of clinical protocols in cancer. Talk about the difference between testing a single agent on its own and adding an agent to a combination that's already thought to have some activity, and how can one hope to tease out the additional benefit from the new element relative particularly to historical use of a simpler combination? And
Mark, maybe you can take that because you are next to the lab. Maybe you can help us with that.
Oh, me?
Yeah.
You, Mark. There are two Marks. It gets confusing. First off, this is a fantastic question, actually, Charlotte. So thank you for asking it. This is one of the challenges we have, is that this is a rare cancer. So we're never going to have trials of 500 patients with 5-FU interferon and 500 patients getting a triplet and teasing out a bit of benefit from a third agent. So I think this is really hard. And another thing is, when you add additional agents together, you often increase the response rate. But in many cancers, we find that giving agents all together versus giving them one after the other doesn't actually improve survival. An example of that is breast cancer, where for years they piled on more and more drugs until they realized that if you give drugs sequentially, the survival is, if anything, better than piling them all together. So, I think this is a real challenge. How many drugs do we put together, and how do we tease out the benefit of additional drugs when we don't even know how effective a 2-drug combination is versus a 3-drug combination? So I can't say that I have a totally straightforward answer for you. I think if the response rates are high enough with the triplet, you can really have the sense that maybe the third agent is adding something over the response rate of the other 2 drugs. Sometimes you can time it by introducing a third drug at a later time point to see that there's a change when you add that third drug in. And I think that's a strategy that's being used by Dr. Kaseb here in one of his studies. But I think this is going to be a challenge for us as we try to evaluate new combinations. Hopefully at some point we'll have drugs that are effective enough for this cancer that we can study them alone before mixing them in with triplets.
And I would like here to clarify one important thing, because you brought up very important, Dr. Yarchoan, is that I know we don't want to go in the lingo to be fair and respectful to our patients, but ultimately the aim of any therapy is to, and on purpose we didn't want to bring this up because we don't want to go in the nitty-gritty, but ultimately any therapy is to enhance survival. We want to make sure that we're remaining alive, we're doing well, we're kicking life, and we're doing as much as we can. That's really why Dr. Yarchoan was bringing in the survival component. Yes, we like tumor to be smaller, we like them to be better, we like them to be disappearing even, but ultimately the final answer, are we living or not? I don't want to touch this because it becomes a little bit dicey, but please, I want you to look at it from the broader perspective. We want to kind of like have the patients just carrying on in life, despite the cancer is there. It's not anymore about beating the cancer. We'd like to get there one day, but at the moment it's living with the cancer as long as we can extend life and really make sure that people are doing well in that regard. That's really what Dr. Yarchoan is referring to. But anyway, back to you, Dr. Firth.
So I'd like to pick up a question that actually comes from one of our foundation's co-chairs, Marna Davis, and I'm going to frame it a little bit differently than Marna put it. She asks, what do we know about fibrolamellar that could possibly lead us to future clinical trials? And I think one can ask that even in a somewhat more specific way. We've known now for about 7 years about a major driver of fibrolamellar, the fusion gene that activates protein kinase A. And we've known increasing amounts over those 7 years about consequences of that in fibrolamellar cancer cells. So what do we know from the science that's been done, harking back to an earlier question, that could lead to future clinical trials? Are there examples of that already in the clinic? And how do we really build on the scientific infrastructure and the basic discovery about cancers in general and about fibrolamellar in specific that lead us to ask the right questions in the clinic?
No, that's a great point. I'll touch on that myself, but then I'll pass on a very important part of the question that Mrs. Davis asked, and I will ask Adi to help us with that. But to begin with, the fusion that you're referring to, Dr. Firth, number one, yes, it happened in fibrolamellar. Yes, it can be a very important driver to tell us what we can and target and where to. We have to remember it's not necessarily purely specific to fibrolamellar, but it's important to really use it as a platform to where the trials can go from. And if anything, one of the efforts is to really try to look at what are the things that this fusion can trigger and go after them. We have done several trials. Some of them we already published, actually many of them were published already, and we try to target item after item. And if anything, we discovered that we have to go after many items at the same time. And that's really what's going to come to is how we can target all at the same time. So this will need, again, the collaboration between the scientists and the clinicians to go to that. But however, I would like to carry on a very important point, which is what Ms. Davis is asking us. What the fibrolamellar would possibly lead us to future clinical trials. In other words, how can we even the foundation help in that regard? Ali, your thoughts on that?
It's an excellent question. I think we're at the precipice of really understanding more. And I say that because I think there are multiple avenues that are currently under study. I think, our general go-to now is some combination of chemotherapeutics or immunotherapeutics. I think from the chemotherapeutics perspective, what we do know is that these tumors do respond to select chemotherapy agents. We know that from prior trials on which patients were enrolled. We know that from ongoing work from those on the line and treatment of children with various agents. And I think we'll learn even more about that as we look, as we mentioned before, retrospectively at what both adults and pediatric providers have done in the past and understanding what percentage of time patients have responded to standard chemotherapeutics. So I think that is a known. And then I think the question is how we can build on that known while folks in the laboratory are actively studying new and novel agents. So I think there's that chemotherapy known. I think that we're starting to better understand that the immune system does most certainly play a role in this tumor. I think we don't necessarily understand it with granularity yet, and yet much of what we're studying, for instance, Charlotte, you brought up the triple therapy, the use of interferon to stimulate the immune system, nivolumab, vaccine trials. There are signals that we've seen, and there's evidence to suggest that the immune system does play an important role in this tumor. And so there's a whole avenue of exploration there. And then, of course, to the fusion protein itself, if there's a way to target it, the downstream elements, and or if there's a way to kind of think about these buckets of therapy and combine them in a meaningful way. I think we have pillars of what we know. And as we wait for the laboratory to find even more novel agents, we can at least capitalize on what we do know and work on combined therapies in that realm as a short-term goal.
Thanks, Dr. Ali. Back to you, Mark.
We don't have any pending questions from the audience, so I'll throw out one of my own, which I've wondered about for many, many years in all kinds of contexts. And it's the question of, as one does learn more about the science underlying a cancer and learns more about the disease in general, what becomes the threshold for when one is ready to go to patients and ask them to actually be part of the process of seeing if an idea is a good idea? So is there a defined way to go from the idea to the human study? Do you have to go through an animal study first? Can you go with a good idea if there's a definite rationale? What's the thought about the ease with which we're ready and feel comfortable in asking patients to take part in the science themselves by putting themselves in the process and on the line?
That's a great point. Maybe Dr. Kaseb, you've done quite a bit of that, so maybe you can help us with that.
Yeah, it's a great question, Mark, and it really revolves around two main factors. Number one, our current understanding of how those agents or agent, if it is a single one, can affect the tumor biology. So if we have good understanding of that, we cannot rush to a clinical trial without basic understanding and the expectation that this drug or these drugs combined will have a signal of activity. Number two, also the safety aspects, how safe it is to combine, these drugs. If it is really something novel and they haven't been combined before, then you have to do it in an earlier kind of clinical trial setting, what we call phase 1, with more frequent monitoring, pharmacokinetics, blood sampling, to make sure the drugs are not interacting with each other in a bad way and hurting our patients. However, most of the drugs that we are currently using for fibrolamellar are established drugs, and that's an important aspect to our combinations. Most of the combination strategies now going on are actually based on safety and efficacy coming from other cancers. So that helps us. So the next remaining question is, why do we think this combination is going to help our patients? So it has to be spelled out very well in our protocol to our IRBs and local approving agencies, to the FDA applications, and more importantly to our patients, to explain to them in a simple way why we think this combination is going to work for them, and we can explain that to them based on our own experience or derive that from published data.
Great, thanks so much, Ahmed. Back to you, Mark.
I actually had a question for Paul, and he seems to have dropped off my screen. I don't know if his internet connection dropped.
I'm not sure what happened, but anyway, we'll wait for him. But
am I correct that he's no longer on the call?
No, he's not on the call, so I'm not sure where it is.
So I'd like to pick up a point that our friend Tom Stockwell has raised, and it's again a very general one about clinical trials. What are the relative advantages and disadvantages to having trials involving multiple centers from pretty early on versus taking an idea at a single center and then expanding out later? What are the thoughts about the kind of the relative efficiency of those two approaches? Ali, is that something you'd like to talk about, because you've been involved in both kinds of trials recently?
I'm happy to. It's a really good question, and I think it's something that we think about. I'll give an analogy to how we think about that, even institutionally. You know, again, when we think about rare cancers, we always think about feasibility of individual trials and prioritization. And by prioritization, I mean, we often, when we're opening clinical trials for a single disease, even at our site, tend to prioritize which trials we think have the greatest scientific promise and which we think will have the greatest both tolerability and attractiveness from the standpoint of patients, the ones who we're asking to enroll. And so I think the same thing holds true for the question you're asking. I think we can only learn from large patient numbers. And so, and you can only accrue trials quickly from having them open at more than one center, usually. That being said, you know, so I think there is a lot to be said for establishing small consortiums of multiple institutions. It's much easier to do that kind of trial when you're repurposing existing agents or when you're utilizing a drug that's readily available from a company, for instance, that more than one institution can easily obtain. So I think in that context, it makes a lot of sense to have a trial open at more than one institute to rapidly accrue patients and to really reach a conclusion sooner than later, so you don't waste a lot of time keeping a trial open or continually studying something that may not work. The converse to that are trials that rely very heavily on a specific scientific technology or something that only one site can really do. And so there are novel therapies, vaccine therapies, some immunotherapies that can only reasonably be conducted at one or two institutes, or sometimes only one institute, because you want, when it's an early phase trial, you want to assure that all the safety measures are in place and that the technology and the technique that's utilized for the particular treatment is sound and uniformly conducted. So I think for things like that, sometimes we're bound to one institute or one or two institutes. But it's really only in those specialty type trials that I think that makes full sense. When we have readily available agents, the more you can accrue and at the more sites you can accrue at, the faster you'll reach your conclusion.
Thanks very much. Ghassan, let me ask you if you'd like to help moderate a question about some of the specific trials going on currently for fibrolamellar. I know we didn't want to do deep dives on that, and we actually have a lot of information on the website about those trials, including interviews with all of the panelists who are still on at the moment. Could you find a way to have us talk about that without getting beyond the language of English and
yeah, sure. No, by all means. Actually, I respectfully answered privately to Ms. Davis, because we want to make sure that we don't help create any sense of compete that might be perceived one way or the other. And actually, in preparation, as you know, Mark, we said clearly, my job is actually to help promote, for example, Dr. Kaseb every way he can, rather than me promote Memorial or what have we. This is the aim of showing the collaboration tonight. Nonetheless, you're absolutely right. The clinicaltrials.gov will be an appropriate platform to show any clinical trial that's available. Please be assured if, for example, one of us don't have a certain trial, we'll contact our colleagues and we'll tell you maybe go or reach out to our colleague to kind of, bring another trial. At the moment, most of the studies are driven by the two drivers that actually Dr. O'Neill brought up. Number one, targeted therapy, along the same line Dr. Firth you brought up, which is the targeted aimed therapy for the activation of diffusion. And we have seen some nice work in that regard. And also the other one is the checkpoint inhibitors, or what we call immunotherapy. And of course, between the target, which is like a lock and the key, and the immunotherapy, which is to activate the immune system against the tumor, we think that this could be an important combination. Add to this, I would like to at least point one note, which is not necessarily, again, and please, I'm being very careful and respectful to everybody because the aim is not compete, but I would like to acknowledge the work that Dr. Yarchoan is doing in regard to the vaccination for the Fibrolamellar. It's not the only thing, but it's a good venture in the right direction. And on top of that, to go back to the question that you brought up, what's happening in the lab? One of the very important things that we are working on is how can we bring a CAR-T program for fibrolamellar carcinoma? Because I think all of us in the foundation, we don't want anything less than a cure. We spoke about many things, but ultimately we want nothing less than a cure. And if anything, at this point in the effort, we're collecting biopsies, we're analyzing them at an incredible speed to be able to define what we're going to target in regard to a CAR-T, which means, in other words, we're going to make the immune cells of the patient smell the cancer and kill the cancer until the last cell and really get rid of it and be cured from it. This is going to take time and effort, but if anything, I would like to comment on, and forgive me, Mark, I'm going to jump into the question of Tom Stockwell. Tom, I love your question. It's very important. You're right. It's better to do a large trial, all of us together, rather than do small mini trials here and there. And this is really, as you noticed already, to be fair, this is what we're trying to aim at. We have done a lot of efforts to really put the trials together. And I think maybe, yes, we'll do more. And this is when I go back to the foundation and say, maybe you can even enhance further the cohesion among all our institutions to be able to bring those joint trials that will run all over the world.
Yeah, thank you. I wanted to pick up on a point that is raised in Charlotte's next question. And also I'd like to talk about, it's in a slightly different context as well. But let me maybe start with Paul on this. So there's an opportunity to build on really 65 years or so of history of work in chemotherapy in combinations, where one has a whole battery of drugs that are already approved for various cancers, and there are different strategies to combine them and try to find something that's effective for your particular cancer. And I know that's something you've done a lot of work on yourself in fibrolamellar and other cancers. There also is now, and maybe we'll throw that to others as well, a new set of types of drugs which are different from classical chemotherapy in that they tend to be much more specific to an underlying mutational background, the so-called targeted therapies. And what's really the activation barrier in each case to, on the one hand, put together the broadly active agents in a way that really works for your cancer? And what's, on the other hand, what do you do when you have maybe lab data that suggests a particular targeted agent that's been approved for cancer A could cross over to cancer B? What are the thresholds to actually be ready to take those kinds of combinations or targeted approaches to this particular patient population with fibrolamellar? Would you like to
Yeah, if I'm sorry, go ahead. I didn't mean to interrupt. If I understand you correctly, when do you try something that hasn't been FDA approved for this, which is most things, or there's not an available clinical trial, or there's no, there is no standard of what do you do? In what scenario does that make sense? And, one of the things that I think about all the time, and I'm sure parents and families think about all the time, is, are we over treating someone or doing more than they need and therefore have toxicities that maybe are unnecessary or even dangerous, or are we undertreating and letting the cancer win the day? And that's always a constant struggle. I remember someone saying, when I was in training, I'm okay with someone, I understand someone dying from cancer, but I don't want them to die from toxicity from the treatment I gave them. And I always thought to myself, well, isn't that the patient's decision? It is very hard for patients who have no options. And if they're on palliative care or they have no other choice and they say, hey, I'd like to try something. And you say, well, there's no idea if this is going to help you. It might actually hurt you. It might actually make things worse. And that's a very tough decision. It has to be made, with lots of people involved outside of yourself. And, that gets back to the whole idea of having a multidisciplinary team, lots of people to look at it. And of course, incredibly close follow-up, as Allison had said. You got to really stay on top to see what sort of toxicities might emerge and what you can learn from that. But anyway, yes, we do get a lot of great information nowadays, and it's always hard to know which of those are going to be applicable or not, but it's better than no information, right? Where there'll be a list of drugs that such and such lab says these are going to be effective, these are going to be synergistic, work together, and these will be ineffective. And we're not quite to the point as our infectious disease colleagues where they get a lovely panel of which antibiotics work and which don't. We'd love to be at that point. But we are at the point where patients are desperate and they don't know what to do next. And they've, relapsed many times and they have tried everything you can think of. And they said, well, I'm willing to try something that may not be proven. I understand there might be unforeseen consequences, but I'm willing to try. And I think that's where the collaboration with the patients, making patient-centered care comes from. If they have to be aware, you don't know what you're doing, but you may help yourself, you may not, but you're certainly going to help other people in the future. Our obligation is to publish that data, to collect that data and publish that data to see what we can learn.
Yeah. Let me throw the flip side of that maybe to Mark Yarchoan. So we're now in an era where one can talk about targeted pharmacology based on biochemical pathways that we know are active or less active in particular tumors, and maybe even some genetic information about what aspects of those pathways the tumor most depends on to survive. And similarly, on the immune side, we have some fairly general kinds of tools now that have emerged, like the immune checkpoint inhibitors, and are starting to move towards more specifically targeted tools, as in the vaccine trial that you're leading at the moment. So what's the threshold for bringing some of these kinds of targeted approaches to patients? And how do you see the most effective way to learn about those in the clinical trials? What do you do in a trial to make sure that you get information out that helps justify the sacrifice that any patient makes of taking the risk to take an agent that's never been used for his or her cancer before?
Yeah, there was a lot in that question. Let's pull it apart a little bit. First of all, the patients I've met with fibrolamellar cancer, I have to say, are just the bravest people I've met, right? Because they are on this course without a lot to go on. This is a rare cancer, and every single treatment that they go on is almost like a trial, because we just don't have hundreds of patients and thousands of patients to base our decisions on. I think one of the things you hit on in your question is, how do we learn from each patient on a clinical trial? I'll just answer first of all by saying I think it's wonderful that patients are participating in trials, because it's the only way that we're really going to make big progress in this cancer, is for patients to risk their lives for the knowledge of others, really. Now, we know patients would not sign up for trials if they weren't getting something that they thought could help them. We understand that, but it's also just so important for the field and for all of us. We're all in this cancer together, and those of us on the panel, I know, go to sleep thinking about this cancer and wake up thinking about this cancer, and look forward to a day when we have better treatments, and clinical trials are so important for getting there. I think it's really important that we build clinical trials that answer specific questions. And so, one of the questions is, do these treatments work? And that's why we need to collect, in many cases, scans to figure out if the cancers are shrinking, but also in many cases, blood and sometimes tumor biopsies to really understand what's happening inside of the tumor and what's happening to the blood. And do we understand why it is that some patients are responding and other patients are not responding? And how do we make the patients who are non-responders become responders? So these are all the critical questions that we try to answer when we see patients and put patients on trials.
Thanks, Mark. Maybe Ahmed, you want to also talk a little bit about the same thing in the context of a question from Tim Ray about the role of immunotherapy in trials. Do we expect that that's going to be, is immunotherapy in some form going to be increasingly important in this as in other cancers? And does it also impact other aspects of the treatment, like kind of surgery you're going to do, or whether you might think about transplants and so forth?
Yeah, so the first question regarding immunotherapy, this is very, very timely, because nowadays, everybody's excited, including, patients and physicians, about immunotherapy potential being in effect, targeted therapy to stimulate immune cells, less toxic, more promising. So we've been learning a lot about this space in fibrolamellar. Initially, we thought that it's a cold environment, it doesn't have a lot of immune cells, activated immune cells, and then eventually we found out, our team and others, about the fact that you could activate them by certain maneuvers. So Mark and his team are working on something in this space. Our team is also working on something in this area to give chemotherapy upfront to stimulate the immune cells and then add immunotherapy. So I think in the next few years, and Paul as well, working on the same area, with checkpoint inhibitor, one of the immunotherapy drugs, Opdivo. So all of us are marching, through that way from different, standpoints and different strategies. So I really think it's an exciting area now, because we have different approaches going on in different trials, using immunotherapy combined with chemo in our case, combined with vaccine with Mark's, study, and also combination of chemo and targeted therapy with Paul and Ali. So all of these are based on observations that we have found in either our patients or published in literature. And in the meantime, we share a lot of knowledge through the Fibrolamellar Foundation, through meetings like this and other meetings, the one coming up end of the year. So those scientific meetings through the Fibrolamellar Foundation really help us, all of us, to exchange messages. And regarding the last question, Mark, you had a question regarding surgery and transplant, correct?
Yeah.
Yeah, so
Does
quickly,
the therapy impact the decision about whether transplants would be appropriate or not for?
Yeah, so that's a very, very hot topic now. Are we doing patients, harm by doing some immunotherapy for those who are potentially transplant candidates? Because in effect, we're stimulating their own immune cells, and then if they end up with liver transplant, we want to have the opposite effect. We want to suppress their immune system to prevent rejecting the new organ. So this is a very, very complicated question. It's been addressed now in clinical trial settings, and hepatocellular carcinoma and in other diseases with transplant. Organ transplant is standard. For fibrolamellar, it is not very standard yet to undergo liver transplant, but it is one of the questions that we have to bear in mind. If your patient has got very small tumor and you have a collaborator transplant surgeon who thinks that your specific patient could be transplant candidate, I would be very careful about introducing immunotherapy, especially very close to transplant, because we don't know yet.
Thank you. We have a couple of questions, one of which is not specifically about clinical trials and future therapy, but really changing the conditions of therapy by the possibility of earlier detection of the cancer, which we all believe is likely to make it easier to treat. So maybe we could ask one of the adult oncologists and one of the pediatric oncologists to talk about awareness of this cancer, which is a little bit different in the two communities, I think. how aware are physicians in general? How do patients come to you? Have they been missed over a period of time? And what is the prospect for actually being able to identify patients with this cancer earlier and potentially at least get them to specific treatments and appropriate treatments for that cancer rather than something else at an earlier point? Paul, would you like to start with that? Because you're certainly hearing from a lot of fibrolamellar patients around the world at this stage.
Sure. I think that's a great one. I think it's very hard to detect this early. There's so many things it could be. As the clinicians know, and we can certainly happy to explain it to the patients as well, right shoulder pain is a common sign of liver cancer. Well, hopefully doctors know that, but how many patients are going to think of that? And they spend time with a sports medicine doctor and they do this and that and the other thing. It never crosses their mind that this could actually be a liver cancer. There's other reasons you might have fevers, might be infection. You know, when we see the dramatic cases, fever, weight loss, night sweats, then we say, okay, abdominal pain. But these are young, healthy people. They are the absence of liver disease. And that's why this is a different disease than HCC. And we can't conflate it or to use the same paradigms as HCC. They don't have cirrhosis, hepatitis B and C and everything else. I think it is difficult for earlier detection. There's no good tumor marker that we have like we do for, say, a germ cell tumor or other tumors where we can do a blood test. What would prompt you to test my 17-year-old twins for liver cancer? Why would you look? Why would you undergo a scan or anything else? For the moment, we don't have an early detection tool other than, I think, as you said, people having some awareness about this. And I hope that would make a difference. But I certainly wouldn't want to suggest that the patients could have or should have been picked up earlier. I think that's an extra burden for families I don't want them to bear because I don't think it's true. And I don't think that's fair to say, hey, you could have or should have picked this up 6 months earlier. When I hear the presentations of these patients, and if my student or fellow had said, hey, this sounds like liver cancer, I would have laughed at them. Said, no, it doesn't sound like liver cancer. You can't do that to everyone who comes in with abdominal pain, vomiting, and a right shoulder pain. But at least it should be on your list. So we don't have a good screening tool as far as I'm concerned right now. These are young, healthy people who aren't seeing doctors, other than letting our colleagues always keep it in their back of their mind. And for the pediatric world, I would say normally HCC is never on your mind. Now with this disease, it should be. There's no other liver tumor that occurs in an 18-year-old really except this.
Thank you. Ghassan, would you want to pick that up or from the adult perspective?
Sure, of course. I second all what Paul has said. And to add to this, I'll take it one step earlier. And I think this is what you referred to also, Mark. How can we screen for? How can we know? And I would say that by definition, early symptoms that are not the typical we see in young age are definitely to be brought in to the attention of the pediatrician, of course, or in the adult life to the general practitioner. And understandably, the general practitioner or the pediatrician will refer to the specialist being in the pediatric age or the adult age. Understandably, so far, we have not seen any inheritance patterns regarding fibrolamellar. Actually, we have some studies and we looked at patients who are like, for example, twins, but we did not see that the twins are both affected by the disease. So we don't have any suggestion at all to screening of families in case anybody is affected with fibrolamellar. Nonetheless, an effort that we are doing in support of the foundation that hopefully will be able to declare public at some point is we're trying to build in a dynamic way to understand how patients do develop the cancer so we can understand better what are the things to avoid or to alter in regard to either environment or regard to anything else. And this is something that our dear patients will hear about worldwide as we continue to evolve it with the help of the foundation.
Thank you.
I think if I can just chime in, I think you said something really important that I'm going to just repeat, which is that this is really not the patient's fault. This cancer, we don't know what causes it. It's very, very hard to detect. And that's often one of the things I bring up with patients when I meet them because they wonder what could we have done differently. And at least right now, the answer is this is just bad luck.
And I think to chime in as well, from the standpoint of awareness, it is, I think in the pediatric world, it's particularly difficult because we are primed, or at least many of the pediatric providers are primed to think that hepatocellular carcinoma, be it fibrolamellar, or the conventional subtype that more closely mimics that occurs in adults, we're primed to think that those things don't happen in children, when in reality, of course, they do. The Children's Oncology Group has really strived to try and bring hepatocellular tumors, apart from the standard ones that occur in little kids, to the forefront, and to encourage study and acknowledgement that they do in fact exist, and that they're probably vastly underdiagnosed. Not only because we don't have them at the forefront of our minds, but because there hasn't been clinical trials that are available for these patients. And so when a clinical trial is available, reflexively, physicians always think, oh, do I have a patient of that sort? Should I be thinking about this diagnosis in any of my patients? Now we have more and more clinical trials for pediatrics in particular for liver tumors in the older age group, many of which are carcinomas, fibrolamellar or otherwise. And I think that is really going to help bring awareness, particularly in the pediatric community, that these diseases do in fact occur in younger children.
Yeah, well stated. We have, I think, time for one last question, and I'll pick one up from Charlotte Welker again. Thank you for so many good questions, Charlotte. And again, I'll slightly reframe it. So the specific question here is, does it matter when in sequencing of therapy one would give an immune checkpoint inhibitor? I'd like to broaden that a little bit and really talk about the general problem in clinical trials, that many trials are initiated, many new therapies are initiated first in very, very sick individuals, in the sense that they've had a cancer that's been treated already with multiple agents, gone through a lot of rounds, and you're testing something in tumors that may have already evolved and been selected to be highly resistant. How much of an issue is that with fibrolamellar, which is thought to be genetically a little more stable than some cancers? And is it something that you think about as you're entering individuals into trials, that what have they seen before and does it matter? Mark, do you want to pick up that? I know you have at least one specific issue there that you and the FDA have debated.
Yeah, well, I think there's a broader question here, which is, does it make more sense to give immunotherapy earlier or later in the disease course? I think that's one of the questions. And when designing a trial, where does it make sense to put immunotherapy? And this is a question that I think is debated in many, many tumors. The earlier you give immunotherapy, in one sense, the less chance the cancer has to figure out other ways to hide from the immune system. And so on one hand, it may be earlier, the earlier you give the immunotherapy, the better. There's something called tumor heterogeneity, which basically means that the cancer changes and finds ways to hide. And the earlier you give the immunotherapy, the less tumor heterogeneity you encounter. On the flip side, if you're able to give other therapies first, sometimes you can break up things in the tumor that make immunotherapy not work as well. And you can also cause damage to the tumor that the immune system can see. So, so there are advantages and disadvantages to giving immunotherapy earlier or later. And it's something we think about when we write clinical trials. Do we require patients to get other therapies first or not? And there's no straightforward answer. I think we need more research to answer this question. I think we're going to learn from other tumor types. So, for example, in tumor types where immunotherapy and non-immunotherapies are approved, there are trials going on where one is given first or the other. And we'll be the beneficiaries of those trials, I think.
Great. Ahmed, maybe we'll give you the last word if you have anything to add to that, because I know the trial that you're most involved with really is looking at a staged process of giving a free drug, and actually looking in the middle to see what the effect has been before you add the third.
Yeah, so it goes along with what Mark just said, in terms of some of the tumors, when you start with certain therapy, definitely chemotherapy, you can induce some cytotoxic effects, meaning cell killing, and that can induce new antigens. And this in itself could be attractive to the immune cells to infiltrate the tumor. So that's what we have noticed, and our study is designed that way, to do a biopsy and then chemotherapy for 8 weeks and another biopsy to test this concept and make sure that there is infiltration of the immunotherapy before you start with the infiltration of the immune lymphocytes before you start immunotherapy. So that's one approach, and all the participants here have also other approaches. You just have to put a strong rationale for it and execute a good plan to test it and make sure that patients are fully understanding, because in the end of the day, we're all trying to help them.
Thank you very much. I think it's time for us to draw to a close. I will toss this back to Ghassan just for a minute to let him officially thank his colleagues on the panel. And I don't know if John wants to have a last word as well. Before I do that, I'll just give you one little bit of advertising. It happens that the next discussion in this series is actually going to be about immunotherapy. So we don't have the date fixed. We're still
popular topic, it would seem.
Yeah, a little bit interesting. It'll be in June, and I can promise you there are some two really outstanding panelists who I'll talk with in that particular event. Ghassan, thank you again for sharing this, and let you say goodnight to folks here.
Oh, of course. Well, thanks so much, Mark. And first and most important, I'd like to thank the foundation for the opportunity, and especially a lot of work behind the scene. I would like to thank, of course, Mrs. Davis. I would like to thank John, Sandy, Mark, yourself, and many others, Megan, and many other colleagues. On top of that, I would like to, of course, thank our dear patients and loved ones for joining us tonight. I hope we were of any help tonight to help alleviate some of the concerns or anxieties that come up with those questions. And of course, we remain all readily available at any time to answer any questions. And of course, we all can be reached out through the foundation. And of course, for most importantly, to thank again our dear colleagues. And I would like to again thank Dr. O'Neill, Dr. Kaseb, Dr. Yarchoan, and Dr. Kent for their involvement tonight. And I pass it on to John for the final goodbye.
Yeah, well said, Ghassan. I can't top that for sure in terms of the thanks. The one thing I will say in terms of for everybody who is participating, the patients and caregivers here, you will be getting an email from us, kind of a survey on this, but it also gives you the opportunity to say, what other topics are you interested in? And if you didn't have a chance to have questions answered now, it also gives you an opportunity to pose more of those too. So please look in your email as a thank you for being part of this. But again, it's so important for us to hear from you as to what you think the important things is for the community as well as yourself to hear. So please look for that. And we'll say good night to everybody, or good day wherever you are in the world. And thank you for everybody for a great, great panel. I think it was really, really informative. So thank you and good night.